Very good. Thank you so much. Please, everyone, take your seats. I'd like to introduce our next speaker. Our next speaker is our very first keynote speaker, Dr. James Fagin. It is no mistake that we chose Dr. Fagin to be our lead keynote speaker. Our keynote slots are the most important slots we have in the World Congress, and so to have Jim as our lead speaker was really no mistake. Jim, as you know, is the chief of the endocrine service and a member of the human oncology and pathogenesis program at Memorial Sloan Kettering and professor of medicine at Weill Cornell Medical College in New York City. He specializes in the treatment of patients with thyroid cancer, with his lab focusing on thyroid cancer genomics and on an understanding of the biology of these tumors. He's won the Merck Prize from the European Thyroid Association, the Sidney H. Ingbar Distinguished Lectureship Award of the American Thyroid Association, which he has also been president of, and the Clinical Investigator Award of the Endocrine Society in 2014. From those of us who have heard many of Jim's presentations over the years, I personally would segregate the learning that I've achieved from Jim in three main areas. The first, kind of clarifying the BRAF versus RAS domains within PTC, which was a step forward in my understanding. Further clarifying beyond the genomic work on routine PTC, the genetics of advanced thyroid cancer. And then finally, the really groundbreaking work on RAI redifferentiation, starting in the lab with mice, all the way through with a clinical trial. And in speaking with some of his colleagues down at Memorial, I wanted to get a little bit of a personal aspect of him, knowing some of his laboratory work and his clinical work. And I think we understand Jim as a very kind mentor, especially to his junior colleagues, and he gives freely to them, and importantly, most preciously, the thing that is most precious for them, which are his research thoughts, which he then gives them and allows them to move forward with their career, and he does this generously. So we'd like to introduce Jim Fagin, our first World Congress keynote speaker.
Thank you very much, Greg, for those really kind words. Fortunately, you've just heard a terrific symposium on differential diagnosis of thyroid nodules, so a lot of the groundwork here for early stage disease has already been done for us. I'm going to jump right into this. Before I do it, just to show you my conflicts. And as I mentioned there below, it's certainly impossible to discuss the work of many different groups that have participated in and made important contributions to our understanding of the genetics of thyroid cancer. So I've had to pick and choose, and for those that I'm not covering, I ask for your apology. So as many of you have heard already, because the data on the cancer genome atlas for papillary thyroid cancer is now 3 years old, we understand this disease to be one of the genetically most simple types of thyroid cancers, in that if you look at the overall mutation burden based on studies for which there has been whole exome sequencing performed, papillary thyroid cancers rank among those that have the lowest mutation burden. And as you'll see in a minute, this is quite consequential in how it helps us understand the relationship between the driver mutations and the genes that are expressed as a consequence of that, and the ultimate histology and biology of these tumors. So in a simplified version, what we learned in terms of the drivers of this disease is shown here. The most common mutation, as we knew even prior to the TCGA, are the BRAF V600E mutations. They're present in about 60% of all papillary thyroid cancers. Second to these are mutations in the 3 RAS genes, with NRAS being the most common. And then third in frequency are the receptor tyrosine kinase fusions and other fusions that activate primarily oncogenic kinases, and RET being the first oncogene discovered in thyroid cancer, but many other receptor tyrosine kinases are also involved through these chromosomal rearrangements. Now I'd like to spend a few minutes talking about the relationship between Ras and follicular neoplasms. These here are listed, are the mutations of Ras in the TCGA study, and most of these were actually follicular variant papillary thyroid cancers. So if we just take a step aside for a moment and think about follicular neoplasms more broadly, and I'm going to actually use the pointer on this screen here. If we look first on top, these are the follicular adenoma, follicular thyroid cancer transition, and here below are those that have nuclear features of papillary thyroid cancer but have a follicular pattern growth. You've just heard from the prior panel the new definition of these non-invasive follicular tumors that have papillary nuclear features shown here. Another variety of follicular variant papillary thyroid cancer that is encapsulated are those that do have invasion. They have either vascular invasion or capsular invasion. And there is a genetic study that I'll discuss in some detail, which is a subsection of these tumors or subclass of these tumors that I'll describe in more detail. And finally, you have the infiltrative follicular variant papillary thyroid cancers. And these actually behave more like conventional classical papillary thyroid cancers and actually also resemble these tumors from a genetic standpoint. So one of the common features is actually the presence of Ras mutations being the dominant lesion across all of these entities. We've known this for a number of years, but it's certainly one conclusion that we can reach, and also based on what Yuri Nikiforov just mentioned, these are tumors that actually retain the architecture of the normal thyroid follicle, tend not to give nodal metastases, and they are found across the entire spectrum of these entities. Second is the rearrangements of Pax8 PPL gamma. These two discovered many years ago, first by Todd Crow, initially in follicular thyroid cancer, but since then they've also been found in the other histologies that are shown here. I'd also like to point out something that's really an important differentiating factor. Another mutation that is seen on occasion in these tumors is a mutation of BRAF that is not the BRAF V600E mutation, but is a BRAF in another codon, a mutation in another codon. And the biochemistry of this has been worked out by Neil Rosen and his colleagues at Memorial Sloan Kettering, who have shown that these tumors actually signal like RAS mutant cancers. They signal as RAF dimers, whereas BRAF V600E signals as RAF monomers. And they have a different degree of activation of the MAP kinase pathway, with the RAS mutant tumors activating to a lesser extent and the BRAF to a greater extent. So this particular mutation, for instance, would predict to not respond to RAF kinase inhibitors. So it's really important both biologically in its association with follicular pattern neoplasms, and also therapeutically for those patients with advanced disease, they would not respond to RAF kinase inhibitors. The other sort of fusion that has been found in association with follicular pattern neoplasms, or these THADA fusions, originally discovered through recombinations of follicular adenomas many years ago, and that recently, in a very nice study, Nikiforov's group found a very interesting mechanism by which these mutations induce transformation, which is by activating through a complex pathway, the activation of the IGF-1 signaling pathway. They indirectly sort of stabilize the transcript of IGF-2, and that results to IGF-1 signaling activation. And finally, something that I will return to at the end of my talk. In this study, which was a whole exome sequencing study done by a group in Korea, they found recurrent mutations of EZH1, which is an important epigenetic signaling protein in follicular adenomas, not in follicular carcinomas. And as I'll describe later, these mutations have also been found to be highly predominant in autonomously functioning thyroid nodules. So now let's return for a minute to the papillary thyroid cancer TCGA study. And as Greg mentioned, one of the insights that was obtained from this study was this distinction between tumors that behaved like BRAF mutant tumors versus those that behave like RAS mutant tumors. The distinction was essentially made by comparing their gene expression profile. So this is the RNA sequencing platform, which is shown here. And essentially what was done is to try and see to what extent the gene expression profile tracked with the presence of BRAF mutations, shown here, or RAS mutations, shown here. And based on this, there was a BRAF-RAS score that was developed, and tumors were defined as either resembling BRAF tumors or resembling RAS tumors. And now what I would like to show you is those lesions that are shown in follicular neoplasms, where do they fall within this overall spectrum? So first I mentioned the non-BRAF V600E tumors. So these are the mutations that have mutations like BRAF K601E, which signal like RAS, and you can see in this spectrum, their gene expression profile is also RAS-like. Similarly, the PAX8 PPR gamma rearrangements, for mechanisms that we don't understand, their gene expression profile overlaps with that of RAS tumors. And finally, the theta fusions, which are also follicular pattern neoplasms, also fit within the RAS spectrum. So you see this overall consistency between genotype, gene expression profiling, and whether tumors resemble more RAS-like tumors or BRAF-like tumors. So next what I'd like to do is to just remind you of the association between these driver mutations and the expression of genes that are involved in thyroid differentiation. And this has been discussed at this and other meetings previously. I'll go through this relatively briefly. These genes that are bracketed by this arrow are genes that are involved in iodine metabolism, thyroglobulin, thyroperoxidase, the sodium iodide symporter, and others. And what you're looking at here are tumors that are actually, each one of these columns represents the sample of one particular patient, and green represents decreased expression, red represents increased expression of these proteins. And what you can immediately see is that the tumors that are BRAF are essentially associated with decreased expression of iodine metabolism genes, which would predict, of course, that they would respond less well to radioactive iodine therapy. Here in this middle panel, what you see are the tumors that have either RAS mutations or receptor tyrosine kinase fusions, and here you can see that the expression of these genes is preserved. But one of the surprising aspects of this data was that not all BRAF tumors were alike. There was a segment of these BRAF, a cluster, that was essentially associated with preserved expression of these thyroid differentiation genes. And we don't quite understand why that is, but one factor that's receiving a lot of attention is that the expression of certain microRNAs, and microRNAs are small non-coding messenger RNAs that can affect the expression of multiple other messenger RNA targets. These track differentially with these two different groups. And there is some early data from Pilar Santisteban in Spain and other groups suggesting that these could be causally related to these two differential groups, but more needs to be done in that area. So summarizing what I've just described so far, if you now try and connect all of this information, and we think of tumors that are either driven by BRAF, by receptor tyrosine kinase fusions, or by RAS mutations. And they, as we've discussed already, they have different histologies, these being more classical papillary thyroid cancers or tall cell variant in the case of BRAF. The RTK fusions, again, have a classical histology, whereas the RAS have follicular variant. In terms of how they signal, and this focuses specifically on signaling in the MAP kinase pathway, one of the important elements in understanding this pathway is that once it's constitutively activated through a mutation in one of these genes, they actually put in place a whole cascade of phosphorylation events. But there's a mechanism by which this effect is eventually shut off, and this is through negative feedback loops in such a way that if you activate expression of this pathway, there are essentially one of the distal effectors has inhibitory effects upstream. In the case of BRAF V600E, this particular gene signals as a monomer and therefore is unresponsive to these negative feedback loops. This results in a particularly high activation of the MAP kinase pathway and a more profound inhibition of the expression of thyroid differentiation genes. By contrast, the receptor tyrosine kinase genes oncogenes actually also activate signaling, but remain responsive to this negative inputs, and therefore they have a dampened activation of MAP kinase and a relatively preserved expression of these thyroid differentiation genes. And towards the end of the spectrum are the Ras tumors, which these are more fully responsive to these inhibitory pathways, and they have the more attenuated activation of MAP kinase. So there's this reciprocal relationship between activation of MAP kinase signaling and the expression of thyroid differentiation genes. And the proof of principle that these were truly connected with each other is from this study that was led by Alan Ho at our institution, in which when he treated patients with RAI refractory metastatic thyroid cancer with an inhibitor of MEK, which is distal in this pathway, he could restore expression of the sodium iodide syn-protein and hence incorporation of radioactive iodine in metastatic lesions. So there's a number of additional studies subsequent to this that are exploiting this overall mechanism in a way proving that this association is actually causal. Now, one of the ways that we're trying to monitor this is by trying to develop assays that will allow us to detect in tumor samples what is the likelihood of actually responding or not responding to radioactive iodine. And we've taken advantage of the Cancer Genome Atlas data set to try and identify genes that will allow us to do this using a technology which is called nanostring, which essentially is a way of measuring messenger RNAs by using a barcoded probes that I won't get into detail, but essentially if you have the messenger RNA target here, then you have two different probes. One is a capture probe, the other one is the reporter probe, and this has a barcode. And so essentially you can read these different barcodes and you can design these nanostring platforms to be able to simultaneously assess the expression of 200 or more messenger RNAs. And so the way we thought about doing this is first we use the 16 genes that were used by the TCGA to define what they call the thyroid differentiation score, and we enhance this computationally by looking at genes that were either positively or negatively associated with these 16 genes. We call this the enhanced thyroid differentiation score. And to this, we used different markers of the MAP kinase transcriptional output as well as controls for tumor purity and housekeeping genes and so on. So one way in which we've tested this is in a clinical trial using the RAF kinase inhibitor vemurafenib in patients with radioactive iodine refractory metastatic thyroid cancer. And the design of the study is shown here. This is a pilot study. All of these patients had BRAF V600E mutations, and these patients had lesional biopsies done while they were taking, prior to taking vemurafenib, and then while they were on vemurafenib. Ultimately, if these patients restored their radioactive iodine incorporation by measurement of I-124 scans, they received radioactive iodine treatment, whereas if they did not, obviously they were not treated. So what you're seeing here is essentially the performance of this nanostring assay. And so what you're looking at here is this enhanced thyroid differentiation score, with blue being decreased expression of these genes and red being what happened after vemurafenib. And you can see there were 3 patients that we're illustrating here. One patient that had a remarkable increase in the incorporation of I-124 in this metastatic site. This corresponds to this patient that actually had the more differentiated tumor at baseline and had the dramatically greatest improvement in thyroid differentiation in response to the drug. Whereas this second patient that had an intermediate response to I-124 uptake actually also had an intermediate behavior in the assay, whereas this patient barely responded at all, had very profound differentiation at baseline, which was only modestly restored by the drug. So it looks like assays of this type actually could play a role in predicting which patients may or may not respond to radioactive iodine. So let me move on to another important marker that you have all heard about and which relates to these mutations in the promoter for telomerase, a gene that's important, of course, in cell immortalization and aging. What has been found is that in 9% of papillary thyroid cancers, and this is from TCGA, they harbor mutations of this, in the promoter of this gene. In poorly differentiated thyroid cancer, the mutations were present in 40%, whereas in anaplastic thyroid cancers in 73%. Now, these mutations are very strongly associated with either BRAF or RAS mutations. And if you sort of look at the potential mechanisms that are involved, when you have activation of MAP kinase signaling, this results in phosphorylation of this transcription factor called ETS, and ETS, when it's phosphorylated, is able to bind to this mutant TERT promoter and activate the expression of telomerase. And that could ultimately result in cell immortalization, accumulation of additional mutations, and therefore a worse prognosis. But there are many non-telomeric functions of TERT that also could play a role in the biology, and all of this work is currently under active investigation by many groups. Now, importantly for those of us in the thyroid cancer field, the work of Ming Xiaojing's group has shown very clearly that TERT promoter mutations are a negative prognostic marker for papillary thyroid cancer, as shown here. This is for recurrence-free survival, and what he did here is compare patients that had either TERT mutation alone or BRAF mutation alone, or their combination, and you see very clearly that these are associated with worse outcomes in terms of greater progression-free survival. Now, one important point is, as we get more next-generation sequencing studies done, is that when you have deeper sequencing, you can get a sense of what the allelic fraction of these lesions is. What do I mean by allelic fraction? If you think of the early mutations in the disease, BRAF mutations or RAS mutations, they happen very likely in the very beginnings of the clone, probably in the very first cell that is transformed. And therefore, they are carried over throughout the entire life history of that cancer. They're present in every single cell. There's been some dispute by some authors about this, but I think the TCGA made it quite clear that these lesions are truly clonal. What happens with these lesions that probably occur a little later in disease progression? Are they present throughout the entire clone, or are they just present in a subset of the clones? And you can determine that by looking at the allelic fraction, the percentage of mutations in this secondary gene as compared to the mutations in the initial driver mutation, which is what we're showing here. And this is work from Inigo Landa and a group who looked at this question. What he found, just mining the data from the TCGA, was that the TERT promoter mutations were actually subclonal in that disease, whereas in anaplastic and polydifferentiated thyroid cancer, they're clonal. This clearly shows that there's a microevolution that supports this important role of TERT in tumor progression. And I'll return to this topic of subclonality a little later. So let's talk now about the genomics of more advanced forms of thyroid cancer, anaplastic and polydifferentiated thyroid cancers. First, I'd like to quote this very nice study from Kansmer et al., which was a whole exome sequencing study of anaplastic thyroid cancers over 20 tumors, 15 of which were paraffin-embedded tumors. And what they found was essentially some of the mutations that we already knew to be present in this disease, like P53, the key drivers, BRAF, RAS, and others. And then one important strength of this paper is because they are surveying the entire exome, they were able to find relatively infrequent mutations as well. A particular concern with this type of analysis is how deep the sequencing went, and I'll explain in a second why. And what we found, as compared to our own study, is that we did find sort of many commonalities between both of our studies, but we found the frequency of the mutations that we both found to be much greater. And part of the reason is because we sequence in greater depth. And I'll explain a little bit why that's important based on this particular issue. It turns out that anaplastic thyroid cancer essentially are enveloped in a network of M2 macrophages. These are two studies that have looked at this question. The first was by Mabel Ryder. The second is by Dr. Caillou at Villejuif. And what you can see here is that if you stain for macrophage markers, essentially these are widely distributed across the tumor specimen. And this is from the Caillou paper. They very beautifully show how these form a network that envelops every single cancer cell. If you now look at the overall tumor purity of anaplastic thyroid cancers as compared to other tumor types, you can see that in papillary thyroid cancers, the purity is over 70%, but in anaplastic thyroid cancers, only 42% of your sample will actually be tumor cells. So when you're doing sequencing studies, if you're just doing Sanger sequencing, you're very likely to miss many mutations because the power of the analysis is diminished by the fact that the sample is admixed with stroma. We use this approach, which is a next-generation sequencing panel of 341 genes that are sequenced with great depth, 500x or more, and obviously I don't expect you to read all of these, but these are essentially genes that have been already proven to be present in many different cancer types and that we can use as a way of assessing mutational spectrum. So this is the overall data from this study that was a study of over 80 poorly differentiated thyroid cancers and more than 30 papillary thyroid cancers. And I will go through this in some detail step by step. But here what you're looking at are the main driver mutations. These are the TERT promoter mutations. You can note immediately that p53 mutations are markedly enriched in anaplastic thyroid cancers, TERT promoter mutations as well. But I will go through this step by step. First of all, if you look at the overall mutation burden, as you might predict, the mutation burden in anaplastic thyroid cancer is much greater than in poorly differentiated thyroid cancer. But if we just focus on these tumors that can vary considerably in their mutation burden, just the poorly differentiated thyroid cancers, and you stratify these based on whether they were at the median for the mutation burden or below or above, what Laura Bucay did, which was look at these data retrospectively, was to find that those patients that had a higher mutation burden were the ones that had the worst outcome. As I mentioned, BRAF and RAS are also the main drivers in this disease. BRAF mutations are present with less frequency than in papillary thyroid cancer, but they're still dominant in anaplastic and poorly differentiated cancers as well. But RAS mutations are comparatively enriched. And we found some interesting differences between both of these. Well, first of all, when you think of the definition of poorly differentiated thyroid cancer, the one that is widely accepted is the one by the Turin proposal, which essentially defines these as tumors with a solid growth pattern and that have high-grade histological features. Dr. Gossain at Memorial Sloan Kettering has defined another class of poorly differentiated thyroid cancers that simply are defined by the presence of a high mitotic rate and necrosis, irrespective of their growth pattern. When we look back, what we found was that these two definitions of poorly differentiated thyroid cancer tracked differentially as well with the driver mutation, such that those that defined an MSKCC with these characteristics were primarily BRAF mutant tumors. Those that were defined by the Turin protocol are primarily RAS mutant tumors. And the other aspect which was of interest was the fact that these different mutations also dictate distinct tropisms for metastasis, such that tumors that have BRAF mutations have a lesser predisposition to develop distant metastatic disease than tumors that have RAS mutations. The other interesting aspect of our study was the presence of these mutations initially described in uveal melanomas, which are mutations of this component of the translation pre-initiation complex. So this is a gene that's involved in regulating translation of messenger RNAs. And these mutations are recurrent. They happen either in the N-terminal or the C-terminal domain. With this particular mutation here, which is a splice acceptor mutation, is private to thyroid cancer. What do we mean by that? It's not found in any other cancer type that we've been able to identify. And they are very dramatically enriched in advanced thyroid cancers as compared to papillary thyroid cancers. 1% here, over 10% here. The other interesting aspect is that these tumors are very strongly associated with RAS mutations, an extraordinary association. So the presence of RAS with IFACs is actually dramatically enriched in advanced thyroid cancers. And this actually is also associated with worse outcomes, as shown here. So next, what I'd like to do is to discuss specifically some of the mutations that are present in different components of the epigenetic machinery, and specifically of the SWI/SNF complex and of histone methyltransferases. And this is particularly the case in advanced forms of thyroid cancer. So this is illustrated in greater granularity here, where you see these mutations in this particular complex that I explain in a second its function, which is called SWI/SNF, which are present with varying frequencies in both anaplastic and poorly differentiated thyroid cancers, as well as with histone methyltransferases. And if you look at the overall frequency, is actually particularly high, and the combination of mutations in SWI/SNF and histomethyltransferases is present in up to 60% of anaplastic thyroid cancers. So clearly, in addition to these driver mutations, what we're seeing are mutations of genes that regulate the epigenetic control of gene expression. So let me very briefly, just to conclude in the last few slides, explain this. There are two complexes that we're going to focus on. One is this SWI/SNF complex, and the second is this polyclonal repressor complex. These tend to be mutually antagonistic, with the SWI/SNF complex essentially having an ATP-dependent capacity to open chromatin and make it more accessible to transcription factors. So when this complex is functioning properly, it actually allows access of transcriptional factors to regulate the expression of genes involved in differentiation as well as in self-renewal. When there are mutations that disable the SWI/SNF complex, then the polyclonal repressor complex comes in, makes chromatin more compact, and it does through the activity of its catalytic enzyme, which is called EZH1 or EZH2. This introduces modifications in histone residues. It methylates one particular lysine residue on H3. This closed chromatin represses differentiated function and increases the expression of self-renewal genes. So what about this polyclonal repressor complex? I mentioned that the catalytic enzyme is either EZH1 or EZH2. And it turns out that a very important study, in my view, that was published by Calabiro and colleagues in the JCI last year, showed that autonomously functioning thyroid nodules had a remarkably high incidence of a recurrent mutation in EZH1 that increases the catalytic activity of the PRC complex. In a study that was done, this whole exome study of follicular adenomas, there was also a relatively high frequency of mutations in EZH1. One of these, which is common to the autonomously functioning thyroid nodule, and another one, a recurrent mutation at a different site. And then looking back at the TCGA data, I looked at this, and there were actually one tumor, which happened to be a follicular variant papillary thyroid cancer, also had a mutation at one of these sites. So it looks like at least a subset of thyroid neoplasms, mostly early stage disease, are associated with EZH1 catalytic activity increase. So what about the other side of this mirror? This relates to these mutations in components of the SWI/SNF complex. It turns out that these mutations are present in 20% of all human cancers. It's a very complicated complex. There are many different subunits, 10 to 13 of them, and I'm not going to go through each of these, but they actually are involved in many different cancers. So this again is returning to the anaplastic thyroid cancer data. These are the mutations of SWI/SNF in papillary, poorly differentiated, and anaplastic thyroid cancers. Now, if we again look at their clonality as compared to papillary thyroid cancers, as compared to advanced thyroid cancers, you can see that the mutations of SWI/SNF, again, are subclonal in papillary thyroid cancers when they were present in the TCGA and become clonal in advanced disease. When you look at the distribution of mutations in these different complexes, many diseases, such as rhabdoid tumors, ovarian cancers, there's a predominant mutation in one of these individual complexes. What we see in thyroid cancer is that they are essentially, these mutations are distributed relatively equivalently between many different components of SWI/SNF. So we've decided to systematically look at this question, and I'll just show you just two pieces of very early data. The first, in which we engineered mice that have BRAF mutations, but also have loss of function of either ARID1A, SMARCB1, or ARID2, three different components of this complex. And the phenotype is totally remarkable. BRAF mutations that we've shown, and many others have shown before, just give you papillary thyroid cancers. But if you also have loss of either ARID1A, ARID2, or SMARCB1, they result with very short latency in development of poorly differentiated or anaplastic thyroid cancers, and the mice die at a very accelerated rate. And finally, this just show you this early experiment. So the question is, since these genes are involved in loss of differentiated function, what happens then when you lose their activity in BRAF mutant disease? And specifically, are you able to restore differentiation in tumors that are BRAF mutant if they also harbor SWI/SNF mutations? So here you have a BRAF mutant tumor at baseline, that has overall decreased expression of the sodium iodide symporter, thyroglobulin, TSH receptor, PAX8. If you treat with this RAF-MEK inhibitor, you get marked restoration of expression of these genes, as we've shown before. But what happens when you have loss, for instance, of SMARCB1? At baseline, they have even lower expression of these genes, and the RAF-MEK inhibitor is totally unable to redifferentiate them. So I think for the first time, we're seeing that when you have an association of these epigenetic mutations with BRAF mutations, those tumors cannot be redifferentiated. So a question that we need to explore in the future is whether other perturbations might restore that function in the future, which is going to be an area of active investigation for our group. So I think I'll stop here, and I just want to acknowledge the very many collaborators that participated in the work I described today. Just in the interest of time, I'll ask you to actually read this as opposed to mentioning them individually. It is not just members of my own lab, but my fantastic collaborators at Memorial Sloan Kettering, as well as at other centers. Thank you very much.
Jim, thank you very much for a really wonderful, wonderful, clear, and succinct presentation. On behalf, maybe just move over a little bit this way here. On behalf of the World Congress, we'd like to thank you so much for your time in your lectureship. Thank you so much.